Good afternoon, everyone. My name is Marissa Shams-White, a program director here at NCI. On behalf of the Obesity and Cancer webinar series, I'd like to welcome you to today's webinar titled Sleep and Circadian Rhythm Assessment in Obesity and Cancer Research, Current Approaches, Challenges, and Opportunities. Next slide, please. I'd like to take a moment to acknowledge my wonderful colleagues who made this webinar possible. The webinar planning team listed here, as well as the Obesity and Cancer Measurement and Methods Working Group, Obesity and Cancer Webinar Series Planning Committee, and the NCI Transdivisional Obesity and Cancer Working Group. It was a true group effort. Next slide. Before we dive into today's presentations, I want to provide some quick information on the webinar platform. So all lines have been muted on entry and will remain muted for the duration of the webinar. Please submit questions throughout the presentation using the chat or Q&A box and select all panelists. A moderator will then share your comments and ask your questions on your behalf at the end of the presentation. Next slide, please. If you require technical assistance, please feel free to contact the host via the chat panel as shown here on the left, and she'll be happy to assist you. Closed captioning is also available and a link has been provided in the chat box. Next slide. We also wanted to make you aware that today's webinar is being recorded. And lastly, we ask that you take a quick moment to evaluate this webinar via the emailed link provided at the conclusion of this presentation. We really appreciate your feedback, which will help inform our future webinars. With that, let's get started. Next slide. As many of you know, interest in self-report and wearable device-based sleep and circadian rhythm measurement has been increasing over the years, especially in light of growing evidence for the role of circadian rhythm and sleep on various health outcomes, such as obesity and across the cancer continuum. On this slide, I've included 13 obesity-related cancers, highlighting the importance of looking at risk factors that impact obesity. That being said, naturalistic sleep assessment poses unique challenges. We're hosting this webinar in an effort to share what is known about this growing area, guided by leading experts in the field. Next slide. Our first speaker will be Dr. Ken Wright. Dr. Ken Wright is a college professor of distinction in the Department of Integrative Physiology and director of the Sleep and Chronobiology Laboratory at the University of Colorado Boulder. He has published over 125 peer-reviewed manuscripts and has over 25 years of experience in the sleep and circadian fields, has led individual and multi-center and team research grants, and has participated in multi-center clinical trials. His research is aimed at explaining the physiology of sleep and circadian rhythms in humans, understanding the health and safety consequences of sleep and circadian disruptions, such as metabolic dysregulation, impaired cognition, and compromised performance, and applying knowledge gained to develop sleep and circadian-based countermeasures to improve public health and safety. Dr. Wright received his PhD in behavioral neuroscience from Bowling Green State University and completed his postdoctoral training at Harvard Medical School. Dr. Wright's talk will be on circadian rhythm and sleep health biomarkers and impact of circadian and sleep disturbances on obesity risk. Our second speaker is Dr. Susan Redline. Dr. Redline is the Peter C. Farrell Professor of Sleep Medicine at Harvard Medical School and Director of the Program in Sleep Medicine Epidemiology at Brigham and Women's Hospital. She leads the Sleep Reading Center for a number of major NIH multicenter studies, including the Sleep Heart Health Study, has led several large cohort studies, including the Cleveland Children's Sleep and Health Study, and has published over 500 peer-reviewed manuscripts. Her research includes epidemiological studies and clinical trials designed to understand risk factors and outcomes of sleep disorders and the role of sleep interventions in improving health. Dr. Redline received her medical degree from Boston University School of Medicine and her MPH from the Harvard School of Public Health. Dr. Redline's presentation will be on measuring sleep health across the lifespan from a population health perspective. For this webinar, both presenters will deliver their talks and come together for a joint presentation with summary statements and concluding remarks. We'll then hold a Q&A panel moderated by Dr. Sydney O'Connor, a cancer prevention fellow here at NCI. So without further ado, I'll give the floor to Dr. Wright.
Thank you very much. I'd like to go ahead and thank Dr. Shams-White and Drs. O'Connor, as well as the other members of the webinar planning committee, as well as the National Cancer Institute, for putting this webinar together and inviting me to be part of this. So today I'm going to be talking to you about circadian rhythm and sleep health biomarkers. And we can think about how these disturbances in these systems can contribute to things like obesity risk and related to cancer. Okay, and so first, in terms of my disclosures, much of the research I'll be presenting today was supported by the National Institutes of Health. And please know that I do consulting for a number of companies that are developing circadian therapeutics. And I will not be talking about any of those in today's webinar. And so for the basic outline of what I'm going to be talking about today, I'm going to be talking a little bit about some basic principles of human sleep and circadian physiology as it relates to health and how we currently assess things. And then I'll go into talking about physiological and behavioral mechanisms that link insufficient sleep to weight gain and obesity risk. So first, one of the concepts that I want people to get from today's webinar is that our biology is not static across the 24-hour day. All right, many physiological and behavioral processes occur at specific times a day. And when we think about metabolic processes, the circadian clock as well as sleep are really important in terms of partitioning behaviors as well as metabolic processes according to time of day. So for example, the circadian system prepares the body for wakefulness and food intake and being physically active. And the sleep system, as well as the circadian system, alters our metabolic physiology for rest and recuperation. And these systems strongly regulate a lot of the parameters that we think about in terms of the metabolic field. We also know that from research comes from John Hoganash's lab, and Ruben is the first author in this paper, that if you look at 13 tissues in human genes post-mortem, we can see that of about 17,000 genes looked at, about 7,486 of them were rhythmically expressed, meaning that they're different depending on the time of day. And a number of these genes are drug targets, including about 917 of them, including cancer drugs for these drug targets. And so time of day matters is one of the key concepts to take away from this talk today. Okay, so I mentioned human physiology varies across the 24-hour day. So how do we assess the circadian rhythm? So first, we have a master circadian clock that is located in what's called the suprachiasmatic nucleus of our hypothalamus. All right, so the hypothalamus is important for a lot of the behaviors that we will think about in terms of metabolism, in terms of thermoregulation, in terms of food intake, in terms of driving behaviors. And in this case here, it also controls, to a large extent, sleep as well as the circadian system. And so the suprachiasmatic nucleus of the hypothalamus itself controls the rhythmic expression of a number of key markers that are reflective of the timing of this master clock in the brain. And this includes the melatonin rhythm here on the top right. We have the body temperature on the bottom, or middle, excuse me, and then the cortisol on the bottom. And in general, when melatonin levels are high and body temperature is low and cortisol levels are rising, we consider that to be the biological nighttime, right? When our behavior as humans is designed for rest, recuperation, and sleep, not being physically active, not having food intake. And then when melatonin levels are low during the daytime and body temperature is high, we call that the biological daytime. And again, our physiology is very different depending on what biological time it is. Now, we also have clocks present now, we know, through most of our tissues in our cells and our body, including a lot of metabolic tissues. And these peripheral circadian clocks are responsive to changes in our behavior, in our physiology, driven by the suprachiasmatic nucleus. We know that these clocks are present in, next slide, please. We know that these clocks are present in bone marrow, circulating leukocytes, oral and colorectal mucose, skin, liver, lung, breast, ovary, endometrium. So these are all tissues that are susceptible to cancer, and we know that these clocks are present in these tissues. And disturbances of these clocks can actually increase the risk of developing or worsening the progression of cancer in animal models. And then if we look at a number of behaviors that interact with the circadian system, including wakefulness and sleep, and the light-dark cycle, the light-dark cycle and wakefulness, driven by wakefulness and sleep, largely drives the timing of this master clock in our brain, as well as activity and inactivity can also impact on that. And importantly, activity and inactivity and eating and fasting can be time cues to these peripheral clocks. And so if we're eating at the wrong time, we can actually disturb the relationship between these peripheral clocks and the master clock, for example. Okay, so how do we measure the master circadian clock in humans? And so the gold standard biomarker currently for circadian timing in humans is the endogenous melatonin rhythm. Melatonin levels on the right-hand side here are low, as I mentioned, during the day and high at night. And that's true for regardless of which species we look at. And for humans, again, this is the biological night when melatonin levels are high. We have markers of this called the dim light melatonin onset and the dim light melatonin offset that denotes the beginning and the end of the biological night respectively. And the DILMO, for short, is the most commonly used marker of the phase of the circadian rhythm in humans. And now, as its name implies, we have to measure this in dim light, and so that does present some measurement challenges, and we'll talk about that in a moment. And in terms of the timing of when this begins to rise from low daytime levels, there are a number of thresholds that are used typically. And so the most common one is the 10 picogram per milliliter threshold in blood. And because melatonin levels are about 30 to 40% of blood when we measure them in saliva, we use a 3 picogram per milliliter cutoff in saliva. If we also measure the entire rhythm, we can actually also create an individualized onset and offset percentage because there are huge individual differences in the amount of melatonin that individuals secrete. Next slide. And so in general, the melatonin rhythm has been measured in the laboratory and where we can assess either the onset or the entire curve. We can also assess it and that's done in blood or saliva. We can also assess its metabolite in urine, so 6-sulfathoxymelatonin with frequent urine samples. And that could be done in the laboratory or even in the field. And this has been done quite extensively in research in blind patients, for example. And more recently, people have done at-home DILMOs where we give specific instructions to the patients and they can successfully actually measure their saliva levels so we can actually determine the timing of their melatonin rhythm. And under all these conditions, we have to control for lighting, posture, and food intake if we want to accurately assess the timing of the melatonin rhythm. Okay. All right, so now I have control, so thank you. So the next temperature measurement that I mentioned was body temperature. And in general, when we're using body temperature to determine the timing of the circadian clock in humans, it has to be done under controlled conditions. And so ambulatory recordings of body temperature are impacted by physical activity as well as sleep-driven decreases. And so under those conditions, it's not an accurate measure of the timing of the clock or the amplitude of the clock. And so under laboratory controlled conditions of constant posture and wakefulness called constant routines, we can actually assess the body temperature rhythm. And for this reason, body temperature is used less today than it has been in the past because of the necessary control that needs to be done to accurately assess it. And we can measure that by using rectal thermistors, pills that people swallow, and telemetry send out the body temperature to a recorder. Tympanic temperature has also been used, as well as skin body temperatures. And then in terms of cortisol, we can measure this in saliva, blood, and urine. And cortisol is required to be sampled quite frequently to accurately assess it. So for example, every 20 minutes. And so that also presents some challenges. And so again, therefore, melatonin is the easiest so far as if you can control for at least lighting. Now, the issue of biomarkers is something that has become really important in the sleep and circadian fields. And there's been a concerted effort to develop biomarkers to be able to predict sleep and circadian risks to health, as well as to understand sleep deficits and circadian timing. And the efforts for these biomarkers have progressed over time here, and this workshop here illustrates the status of the field at the time and opportunities for the future. I encourage you to take a look at it if that's of interest. Now, currently, in terms of sleep and circadian biomarkers, the most commonly used ones, especially in the field, are going to be looking at wrist actigraphy, right, where we can assess sleep-wake duration. We can estimate sleep-wake duration about 88 to 90% of accuracy compared to the classic gold standard polysomnography, where we record brainwave activity. But this is also very useful because we can measure many days to weeks in individuals. We can then look at the timing of the sleep, the regularity of this pattern, and then we can also use questionnaires to get at sleep quality when we associate it with these particular outcomes. We can also then translate some of these physical activity counts to look at how changes occur in terms of increases in physical activity at certain times of day under certain conditions. And again, the timing of inactivity and estimated sleep. And then we can also utilize these types of devices to measure light exposure. And I mentioned light exposure is very important for that master circadian clock to sync it. And we can then measure various components of light exposure, exposure to outdoor light, to indoor light, as well as things like campfires. Now, there's a number of novel biomarkers of insufficient sleep that have come out onto the research now. And these efforts are designed to, for example, to take one single blood sample to be able to determine whether or not an individual is obtaining insufficient sleep. And so you could think about these developmental efforts to be added to a health panel in the future, a fasting blood sample, for example, that is typically done in the clinic. And so these efforts are underway, and these are the initial studies, examples of initial studies that have started to pave the way towards this adaptation. And then we also have efforts for novel biomarkers to assess the circadian phase. And in this case, again, using one or two blood samples across the day. And these are usually currently using transcriptomics methods, while others for the insufficient sleep have also used metabolomics. And in this case here, these initial efforts under controlled laboratory conditions have shown that with a single blood sample or two blood samples, we can accurately assess the circadian timing of that melatonin rhythm within 1 to 2 hours. And so again, this is great promise to be able to utilize these types of potential biomarkers in large-scale trials in the future. And then we're also developing novel biomarkers of peripheral circadian clocks. So to try to get at what is the timing in the liver, of the pancreas, and other tissues in the body. And in this case here, the proteome and the metabolome have been used with some initial promise to illustrate and understand what's happening with the timing of peripheral circadian rhythms by using markers of those peripheral clocks. Now, I'm going to briefly talk here now about components of sleep and circadian health. And this was initially put forth the concept of sleep health by Dan Bicey, and Dr. Redline will talk more about that later. But the main components are sleep duration, how long, and the American Academy of Sleep Medicine and Sleep Research Society say that the minimum sleep duration to maintain health is 7 hours per night on a regular basis for adults. But it's also becoming more evident that other components of sleep are also very important for health, such as the timing of sleep. Earlier in general is better than later. Sleep regularity, the consistency in the sleep timing, as well as the quality of sleep. And then we have related concepts in terms of the circadian system. The first one is circadian alignment. And this is when the master clock in the brain is timed appropriately to the light-dark cycle, to the school and work environment, as well as to behaviors such as food intake. And then we also have alignment among the peripheral and the master clock, so everything is working as it should in terms of timing. We also have timing of the master clock. Again, earlier is better than later in general. And then we have the same concepts of regularity of the circadian timing and circadian amplitude, which I won't talk much about today. In terms of assessments of circadian rhythm, we can look at chronotype, which is the timing of the master clock relative to things like lights out or bedtime. So this is the timing when melatonin levels begin to rise relative to bedtime. You can see these individual differences, and these individual differences translate to things like chronotype, such as later types have a later timed melatonin compared to their sleep, as do earlier types. And this timing drives behaviors such that these later types are more alert in the evening and more sleepy in the morning, for example. We can also look at chronotype with questionnaires, and this is by Tilla Runenberg. This is the immunochronotype questionnaire, and this is the timing of mid-sleep on free days. So 4 a.m. here is the middle of the sleep opportunity. And you can see here that in young children, we have earlier chronotypes. As we get later through adolescence and young adulthood, we become later, and then we become earlier again throughout the majority of the rest of adult life. And you can see a sex difference here. And so these types of questions in terms of the immunochronotype can also be assessed with a micro immunochronotype questionnaire with just 4 questions that can be easily added to epidemiological type studies. And based upon understanding when people are sleeping on free days and work days, we can generate concepts such as social jet lag. And this is the difference between the middle of sleep timing on weekends here in green versus the weekdays or work days here in red. And this difference here is called social jet lag because people are living essentially in different time zones on the weekdays versus the weekends. And this degree of social jet lag here is also showing an age dependency and some sex dependency. But importantly, this social jet lag has also been associated with obesity. So if we look at overweight to obese range here in BMI, we can see that the more social jet lag, the higher the BMI across this range. We also know that people's circadian timing rapidly adapts to changes in our behavior, such that when we stay up later on the weekend and we wake up later on the weekend, this is also associated with a change in the timing of our circadian melatonin rhythm. So the whole melatonin rhythm is also later in addition to the sleep. And so this is, again, just a matter of 2 days. And so the system is constantly adapting to changes in our behavior. And here I just want to illustrate a concept of sleep health where we can measure this with sleep logs. You can see that in this case here, this is an illustration of what happens to sleeping university students under the stay-at-home orders in COVID. And based upon these findings, we found that time in bed devoted to sleep increased during stay-at-home on the weekends and weekdays. Sleep regularity improved, so people became more regular, and that social jet lag was reduced. And so those are all positive sleep health behaviors under staying-at-home COVID orders. But sleep timing did become later during weekdays and weekends, and that is not thought to be a healthy pattern. So that just illustrates our ability to actually measure things with simple sleep logs. Here, I'm just going to quickly go through here. How does insufficient sleep contribute to weight gain? And when we think about energy balance, we found that insufficient sleep increases our energy expenditure. And if anything, this should lead to weight loss over time. But we also know that insufficient sleep increases appetitive hormones, increases appetite, and increases food intake. And this drive for food intake and the amount of food intake that occurs is much greater than this increase in energy expenditure, and that leads to positive energy balance over time. Now, there's also evidence that physical activity is decreased during insufficient sleep, and this also can contribute to this positive energy balance. So we know that energy expenditure increases by sleep deprivation and sleep restriction in both humans and animal models. And another concept that I want to just show you here is when people get insufficient sleep here in the middle, this is 5 hours time in bed versus 9 hours time in bed on the top, you can see here that people are awake here in the morning during many hours now when their melatonin levels are high, illustrated by the downward triangle here when melatonin levels drop back to low time levels. So the point here is that insufficient sleep leads to morning circadian misalignment. Now, it also changes the timing of when we eat. When we look at post-dinner snacks specifically, when people get insufficient sleep, they eat much more at that time of day compared to other times of day. And this later eating at night has also been associated with obesity risk. And we also know that from research looking at weekend recovery sleep in blue here, that from baseline, we increase these post-dinner snacks. We have a weekend, those post-dinner snacks go back quickly to baseline. And then when we go back to getting insufficient sleep again the next week, we increase those post-dinner snacks. So we have rapid changes in our food intake under these conditions of insufficient sleep. Now, there's a lot of research looking at food intake and sleep duration and how it's associated with sleep timing, sleep consistency, and increases the risk of obesity. And there's a lot of research opportunities here. There's also metabolic consequences of sleep and circadian disorders that contribute to negative outcomes in terms of metabolic function. And what I want to just briefly say here is that when people are under weight loss conditions, when they're caloric restricted here, and we look at what type of weight loss that they incurs over 2 weeks when they're restricted to food intake to 90% of needed estimates based on RMR, we can see that when people get insufficient sleep here in the black dots, they actually lose as much weight as the same group of people when they get adequate sleep, but they lose less fat and more fat-free mass when they're obtaining insufficient sleep than sufficient sleep. And so weight loss under conditions of insufficient sleep is much less optimal because we're losing fat-free mass, which is something we don't want to lose. And then in terms of time here, I'm just going to go ahead and just jump to one summary slide here. We know that circadian misalignment also decreases energy expenditure when we think of things like shift workers, and this can lead to positive weight gain over time if food intake doesn't go down. And there's a lot of research looking at and a lot of opportunity here now looking at food intake and shift workers and what can we do to help improve their metabolic health. Also important, we don't know how much physical activity and at what biological time is needed to maintain the health of shift workers. We have guidelines for day workers, and they may be insufficient for shift workers. So with that, I'll just conclude that optimal function occurs when sleep is adequate and sleep and circadian timing are aligned to the working schedule. The amount and timing of food intake is important. The timing of our physical activity, and we need more research to understand what is the best for that. And we're also important that bright days and dark nights are very important, and I don't have time to go into that, but brighter days, getting more sunlight exposure, dimmer lights in our homes are very important for our circadian health. We know that sleep and circadian disruptions and untreated sleep and circadian disorders dysregulate our physiology and increases the risk of health and problems and disease. And lastly, we know that there's strong associations between sleep and circadian health and other health behaviors such as food intake and physical activity. And so understanding the association between these is important. With that, I'll thank you very much and acknowledge all the students and the funding that has supported this research. Thank you.
Thank you for that really great presentation, Dr. Wright. Next up, we have Dr. Redline. Dr. Redline, the floor is now yours.
Okay, thank you very much for the opportunity and it's to speak, and I definitely appreciate Dr. Wright's overview, which I'm going to expand in. Let's see. Okay, this is. I'm going to get this to work. Okay, I think it's okay. To begin with, my disclosures include NIH grants, a grant from Jazz Pharmaceutical, and a few consulting opportunities, none of which impact the work that I'll be discussing today. So what I'll do is begin with taking now a higher level view of some of the sleep and circadian concepts that Dr. Wright reviewed. In particular, I'm going to really review how we think about sleep across the population. In particular, take a lifespan approach, as well as think about how sleep disorders may differentially affect populations at risk for health disparities. I'll also briefly review some of the links between sleep, obesity, and cancer, as introduced by Dr. Wright. Talk about measurement of sleep domains, again, taking a population perspective, and concluding with some measurement approaches. So if we begin with a lifespan approach, I'll submit that such approach really may help us begin to think about how sleep influences really the physiology and the behaviors that are manifest across our lifespan, and across our lifespan. In particular, think of first in utero, have a mother's sleep itself, a mother's exposure to discrimination and stress may result in prematurity or placental insufficiency that then influences brain and lung development of the baby, that then may predispose that infant to lifelong problems, and brain and sleep health being critical, and brain and lung health being critical for sleep health. We can also look across the lifespan at specific exposures, many of them that disproportionately affect minority and low-income communities. For example, how maternal depression or exposure in the NICU, the neonatal intensive care unit, may lead to babies, influences on the baby, influencing their ability to self-soothe and therefore get sufficient quantity of sleep. Across mid-childhood and adolescence, various neighborhood and household stresses and behaviors likewise may impact the ability of the children to, follow regular sleep routines and get sleep that isn't fragmented, thus have the sufficient quantity and quality of sleep. And finally, in later adulthood and in aging, other stresses, again, including neighborhood and household stresses, partner-related stresses, and other stresses related to insufficient resources may also impact these same sleep patterns that may be further amplified and result in insomnia, sleep-disordered breathing, poor sleep quality, ultimately leading to weight gain, obesity, cardiometabolic diseases, and cancer. And in fact, using this life course approach, we do see that sleep problems begin to emerge in early infancy. So on the left-hand side of this slide, you see data from a birth cohort in the Boston area called Project Viva. And just simply based on maternal report of the infant sleep at 6 months of age of less than 12 hours, which is a short sleep for a young infant, Black and Hispanic twice as likely to have short sleep than white babies. And on the bottom half of the slide here, you see various risk factors for childhood obesity, ranging from sugary beverages through maternal diabetes. And here are the adjusted odds ratios for having that risk factor if you're Black in purple, Hispanic in orange, compared to the reference group of white infants. And you could see, in fact, that the risk factor, these childhood or infant risk factors for obesity include infant sleep, which is as strong, if not stronger, as a disparate risk factor as many of the others. On the right-hand side of the slide is some newly reported data from our study, again in the Boston area, from the SHINE cohort. And here we measured ankle actigraphy to measure sleep-wake patterns in infants at both 1 and 6 months of age. And what we see is the differences in the white bars of sleep duration at night at 1 month. And then in the hatch bars at 6 months, and then the cross bars is the change from 1 to 6 months. Now, sleep normally follows a developmental pattern, and one of the key factors related to healthy neurodevelopment is the consolidation and the increased duration of nighttime versus daytime sleep in infants. And what you see both in the upper and lower panels is that that it's not 1 month sleep, but it's the change in sleep from 1 to 6 months that's actually less, that's smaller in infants from minority families and infants from low-income families. Again, suggesting even early in life, you have these negative influences on sleep related to the social factors. And in fact, we see similar disparities even in older adults. So here's data from the multi-ethnic study of atherosclerosis, a 6-center US-based cohort of individuals studied when they were mean of age 67 years of age. And you see the overall, and here we use polysomnography, actigraphy, and questionnaires to comprehensively measure sleep health. And you could see in the Black versus the white participants that the Blacks were 5-fold more likely to have short sleep duration, poor sleep quality, and daytime sleepiness. Amongst the Hispanic group, we additionally see a much higher rate of sleep apnea as well as short sleep duration. And in the Chinese Americans, we see not only more sleep apnea and shorter sleep duration, but we see, despite the high sleep apnea, a much reduced likelihood of being diagnosed and treated for sleep apnea. And across all groups, the commonality that we see is the short sleep duration. Now, what I'd like to step back and think about what are some of those factors that interact with the factors that I know this audience thinks about when they think about cancer risk factors. So the focus of this seminar is, in fact, how to relate obesity, energy balance, and metabolic dysfunction to cancer. And what I know has been the traditional perspective is to focus on physical activity and diet. But as Dr. Wright mentioned and showed by his 3-legged stool, there's actually very close interactions between activity, diet, and sleep, as well as the fact that sleep, as well as a number of these other physical behaviors are influenced by many factors. Dr. Wright mentioned light, obviously noise, work, school schedule, substance use, even TV exposures, amongst multiple others. And the point for this audience is to recognize that if the goal is to prevent or improve cancer outcomes through improving obesity, we need not to look at one leg of the stool, but comprehensively think about interventions that holistically address these multiple behaviors that include sleep, as well as those inputs that affect sleep. We heard about some of the strong experimental and epidemiologic data that in fact provide the biological underpinnings to underscore the importance of how sleep disturbances influence metabolic health. Just to reiterate briefly, there are experimental studies in both humans and animals where you sleep restrict humans or you disrupt their schedule, bring them into the lab and study them at their biological, reverse their biological day and night, or even expose them to intermittent hypoxemia, an exposure similar to the disease sleep apnea. And you could measure these changes in inflammatory proteins, both by levels and gene expression, downstream organ responses like endothelial dysfunction, dyslipidemia, and glucose metabolism. And then there is a very strong epidemiologic data that have looked at a variety of single dimensions of sleep, like short sleep duration or increased sleep variability, or reduction in restorative stages of sleep like slow wave sleep, SWS, or sleep apnea, showing those risks, those individual dimensions are associated with cardiometabolic health, including emerging data suggesting they also are predictive of cancer. And here is a high-level framework to think about the biological underpinnings of these associations. So what you could see on the left, in fact, the brain, outputs endocrine and autonomic nervous system signals that influence a variety of factors that influence eating behaviors and metabolism, including satiety, the stress axis, pancreatic beta cell function, lipid metabolism, insulin metabolism. And as we heard, the brain itself is influenced by light and other factors, as well as behaviors that restrict sleep, cause sleep disruption, or cause circadian disruption. And now as you turn to the right side of the panel, you could see that, in fact, not only will these disruptions in sleep, which come in many forms, insufficient sleep, fragmented sleep, sleep variability, and line rhythms, influence diet intake, predisposing to higher energy foods, but we also heard the other exposure that often irregular sleep patterns are associated with irregular eating habits. And those irregular eating habits, eating late at night, grazing across the 24-hour period, rather more restricted eating, irregular meals, will further aggravate metabolic functions and contribute to weight gain, even with the same caloric intake, leading to these adverse outcomes. And when we think specifically about cancer, certainly we think many of the mediating factors are through energy imbalance, cardiometabolic factors, obesity, mood, stress, and so forth. But there are these other biological pathways that are triggered by circadian disruption, insufficient sleep, and sleep disturbances, including melatonin suppression, immune abnormalities, inflammation, abnormalities in insulin. And those factors together have been linked to breast, colon, prostate, and endometrial cancer. And then I wanted to also mention another sleep exposure, that is sleep apnea, which, because of the intermittent hypoxia, seems to have a specific effect on stimulating various growth factors through the HIF1 pathway and VEGF, that has been implicated in the incidence, progression, and survival of people with melanoma, lung cancer, kidney cancer, and bladder cancer. Now, Dr. Wright also introduced the various domains of sleep and circadian of traits that we measure, and I just want to briefly review these before we go into measurement. So he provided a wonderful framework for appreciating the importance of circadian rhythms and particular sleep timing and rhythms. And again, that refers to when we sleep, its variation, which includes social jet lag that we heard about, as well as the simpler concept of just night-to-night variability of when we sleep. And our own data has shown that, especially in low-income communities, it's not uncommon for people to vary their sleep timing by 60 to 90 minutes per night. We also think about the strength of our sleep-wake rhythms, and that can be manifest by how active we are during the day and how restful we are at night. And we could actually model those, and that's a very robust, those parameter estimates give us very robust predictors of a variety of outcomes. Going down the list, we think of sleep duration, probably the most commonly measured epidemiologic measure, which often is thought of as average sleep duration, although we know that the extremes of sleep duration, short sleep duration, less than 6 hours a night, or long sleep duration greater than 9 or 10 hours of sleep a night, are associated with a whole host of risk factors, including mortality. We also tend to think about the domain that we'll lump as sleep fragmentation, efficiency, or depth. And this is the continuity and the depth of sleep, and we measure it multiple ways by how frequently we wake up or have mini awakenings or arousals at night, how deep the sleep stages that we get into, because we typically will have both lighter and deeper sleep through the night, how many transitions across stages, as well as how we perceive our sleep quality. There's also a great deal of interest in using the EEG during the night to not only measure these macro architecture measures of sleep stages, but the specific features of sleep like spindles and slow waves that actually right now may be some of our biggest predictors of some outcomes like cognition. And we also can measure and characterize whether people have sleep disorders, which are common population, including sleep apnea, periodic limb movements, and insomnia. And finally, we could characterize the daytime sequelae of sleep disturbances or insufficient sleep, sleepiness, and functional impairment. However, although I listed each of these as individual domains, what's really critical to understand is that these individual domains often don't occur in isolation. Many individual problems may actually drive other problems. For example, sleep apnea may drive sleep fragmentation, or sleep fragmentation may drive circadian weakening of the circadian system. And on the other hand, many of these individual sleep domains also may occur commonly in any given individual because they're each common. So I think we have to understand they overlap, there's interdependence, and that in fact their combinations may in fact better characterize really health and disease. Now, there are really two frameworks that have been recently been used to thinking about multiple dimensions of sleep health, and those are shown in the next slide. The first one was one proposed by the National Center of Sleep Disorders Research, which coined the term sleep deficiency, which was defined as the deficit in the quantity or quality of sleep compared to the amount needed. So it really was, tagging sort of that deficit. More recently, Dr. Bicey has coined the term sleep health with more positive attributes, which he defines as a multidimensional pattern of sleep wakefulness adapted to the individual social and environmental demands that promote physical and well-being. That is, a higher sleep health score is predictive of overall better health. And we could operationalize sleep health as the following. One, it can be thought of using the mnemonic, are you sated? So this means R for regularity. Do you get consistent timing and duration of sleep? Are you satisfied with your sleep? Are you getting good sleep quality? Are you alert during the day? Again, is the timing aligned with your own biological clock? Is your sleep efficient? Is most of the sleep period spent asleep rather than wake? And are you getting sufficient duration of sleep? And in fact, for each individual, you can grade them on the scale, create a composite score, sleep health score, and map that to your overall health. And in some work that June Chung, a postdoc in my group, recently did, and again in the MESA study, multi-ethnic study of atherosclerosis, we used both polysomnography, actigraphy, and questionnaires to create a new sleep health score, and in fact, found that a one standard deviation increase in sleep health, again, that's a positive attribute, was associated with a reduction in your likelihood of dying by 27%, adjusted for many other risk factors. Furthermore, what he did was he looked to see what was driving the individual components within the sleep health score, which, as you can see, included a number of measures of variability, quality, depth, sleep architecture, and timing. And interestingly, the things that were driving it were really consistency in the duration and timing of sleep, actually having an optimal sleep duration, not too short or long, and not having sleep apnea. Now that we think about conceptualizing sleep health, how do we measure it? So commonly and traditionally, we've used three types of measurements. One is polysomnography, which over the years has been miniaturized, and now we can actually easily bring in the home. And in fact, in studies that I'm doing, even in very sick populations, we could mail these very complicated devices, and patients and people can self-apply these electrodes, and we could measure overnight, sometimes over multiple nights, brain waves, breathing patterns, leg movements, and comprehensively assess the multiple physiological features that occur overnight during sleep. Dr. Wright's talked about actigraphy, and again, this is a simple procedure that really translates movements, usually by the wrist, into sleep-wake estimates. And you could see here two sets of data. Each row is a different day, and that nice green pattern is the lack of movement, what we assume or attribute sleep. And you could see we have a really good consistent sleeper on the top, and the bottom one we have a very inconsistent sleeper that, again, those green bars who's changing behavior from night to night. And there's a series of questionnaires that include, questionnaires on sleep disorders and impairment. They include the PROMIS Sleep Bank, overall sleep quality scale, specific instruments to address disorders like insomnia or likelihood of sleep apnea, or daytime sequelae like sleepiness, as well as chronotype. In more approaches, one of them is oximetry, which is a very simple finger sensor to get at hypoxemia. That is a sleep apnea screen that may be particularly relevant, for promoting some of the cancers we spoke about. And this burgeoning amount of wearable data that not only includes accelerometry, sleep-wake measures, but can measure heart rate variability, temperature, and even oximetry that, really, on a daily basis, as well as using your cell phone to capture information on electronic diaries. So here's an example from an NCI-funded TREC pilot study where we programmed in some questions about light exposure that participants several times a day were asked to report on when they were last exposed to indoor light, and what was the light in the room, and the use of electronic devices that was combined with this other actigraphy-based data. So with all these tools and all these domains, how is one to choose? So it starts with, of course, what are you interested in? What do you want to measure? What's the objectives? And And then we have to think about what might be of those tools and approaches, which are most valid. And often we think about a gold standard, or often we don't know what the gold standard is. So we have to understand that bronze standard. Measurement usually means understanding reliability. How representative is the data that we propose to collect, how for that individual, and how do we capture or address night-to-night variability? What are the potential sources of bias, that both differential and non-differential, that could cause us to misclassify our participants or patients? And then how scalable are those results? And then finally, related to scalability, also, what is the burden to the participant as well as the cost? And this table, I just gave an example of how we might go across the rows looking at questionnaires, actigraphy, polysomnography, wearable, and across the domains. And this just indicates what is feasible. It doesn't indicate which measurements may be most accurate or reliable, but a few trends immediately come out. For example, you could measure sleep duration and timing almost by any of these modalities. However, if you use polysomnography, you're only going to get a single measure, but during that night, that measure is going to be pretty precise. On the other hand, if you use a questionnaire, you may be able to get at what an individual really perceives as their average experience, not limited to one night, or maybe not even limited to the 1 or 2 weeks that you wear an actigraph. As another example, if you look at polysomnography, that's the only tool that we could really measure some of those deep measures of EEG that allows us to look at micro and micro architecture of sleep. But because polysomnography is only one night, it gives us no information on sleep weight. So this is the kind of framework one might think about in terms of trying to map what you want to measure, that is along these columns here on the left, according to the tools available. And as I said, the other part of choosing is to understand misclassification and error, and think about the implications of the error in your particular population. So here is some data from 3 different studies: the MESA study, Hispanic Community Health, and Jackson Heart Study. So in the MESA study here, we see in this upper left corner, we see the distribution of sleep duration in the same individuals measured by 3 techniques. And what you could see when we use polysomnography in yellow, we have the shortest, we have everything skewed to shortest sleep duration, whereas when we use self-report in red, that gives us the longest sleep duration, with actigraphy in the middle. As another example, here is data from the Hispanic Community Health Study that plots the actigraphy-based sleep duration on the y-axis versus self-reported on the x-axis, and you could see that not only is there a weak correlation, but it's not linear across the range of sleep duration, such that those who are shorter sleepers tend to have longer sleep when you use actigraphy. That is, they have what we term sleep misperception. They underestimate how much sleep they have, which is particularly common in people with insomnia, whereas those with long sleep tend to overestimate how much they get relative to actigraphy. And if we think about this, and here's some data again summarized across these 3 studies, there is only a modest agreement between self-reported and actigraphy, between sleep duration about 0.3 to 0.47, and there can be both over and underestimation. I also want to say that this may not only vary by where you are in being a short sleeper or a long sleeper, but how you phrase the question. So we've found that when you ask a person, how much sleep do you usually get, versus we ask them what time they usually get up and go to sleep or go to bed and wake up, you get much shorter sleep duration when you ask them to estimate their habitual sleep rather than actually ask them to estimate their usual timing of sleep. As one of my colleagues said, they almost are self-punishing. And another concerning, maybe surprising fact is even when we look at actigraphy and PSG from the same night in the home, we only get a modest correlation of about 0.29 with an overestimate by actigraphy of 45 minutes. Again, that may be because we're really attributing some, inactive motion as sleep that the EEG doesn't confirm. And again, we've reported in several of these studies that some of these estimates also will vary more by certain in certain populations, defined by race or income or perceived sleep disturbance. Nonetheless, each of these modalities, self-report, actigraphy, and polysomnography, actually do report sleep durations that do relate to the various outcomes of interest. So I think here raises the challenge that I'm sure we're going to discuss later, is then how do you choose the modality, and not only the modality, but the thresholds that really allow us to make clinical recommendations as well as public health targets. So, I'd like to now conclude by summarizing that we have numerous objective and subjective measures. I wanted to emphasize that I actually think there's a lot of complementary information from these. We need to really not discard one group over another. After all, self-reported sleep is a patient-reported outcome, and how people perceive their sleep may be to them more important than what we tell them we measure. I also reviewed some data indicating that there are multiple sleep domains and they're not independent. And for this audience, I'd like to really make the analogy to nutrition and in fact give a call out to one of my colleagues, Dr. Tian Huang, who is a nutritional epidemiologist who's now working in sleep. And he's pointed out to me that just like we've been talking about sleep health domains and sleep health scores, for many years, the nutrition literature actually focused on specific nutrients and often have moved then from a nutrient-focused perspective to a more global diet perspective, like the Mediterranean diet, meaning some of the parts may give more information than the individual parts. And I think we could make that analogy to a sleep health score, especially when it comes to tracking population health, over time. And I think where some of the challenges may be is, how we might think about those composite scores as also relating to how we may want to design interventions. Are there a couple of components that are really intervenable that will then drive improvements in other components? And are there specific individuals with disorders where we really need to know measure those components specifically rather than focus on the composite? And then finally, I like to just conclude by, and we're going to talk about this in the discussion session, this is a fantastic time to be in sleep research, and there is this opportunity to integrate and utilize multiple data sources and advanced data processing. So I'd like to conclude and acknowledge some of the studies that contributed data to the study. I have multiple colleagues, but for this presentation, wanted to acknowledge the work of Sean Purcell. You'll hear more about his work soon. Chandra Jackson, Peter James, Xiaoyu Chen, and Xingting Li, and our funders. Thank you very much.
Thank you, Dr. Redline, for that wonderful presentation. I'm now pleased to present both speakers for a joint presentation titled Wearables and Big Data, Future Opportunities in Sleep and Circadian Research. Dr. Wright, I'll give the floor back to you.
Thank you very much. And so I'm going to start talking about wearables and talking about future opportunities for sleep and circadian research. And I'd like to highlight that the identification of biomarkers of sleep and circadian disorders is part of the NIH Sleep Disorders Research Plan. And the goal of this is not only to facilitate sleep and circadian science, but also to be able to utilize these biomarkers for personalized medicine, personalized treatments, as well as look at the risk associated with untreated sleep and circadian disorders of a number of diseases such as obesity and cancer. And next slide, please. I also put at the top here now the outcomes of another workshop that was supported by the National Institutes of Health, and it was a Circadian Health and Light workshop. And in there, we discuss the opportunities for measuring light exposure with wearables, as well as understanding how light is important for health. And there's a lot of interest in this area, specifically in cancer as it relates to shift workers and increased risk of cancer, for example. Next, I want to just highlight another workshop. And actually, thank you very much. I want to highlight the workshop that was run by the Sleep Research Society. And it was a workshop that brought together leading scientists, experts in wearable technologies, as well as representatives from a number of consumable companies that develop consumable wearables. And based upon the outcomes of this workshop and review of the existing research literature, they came out with the following statement and conclusion. And so it was their position within the current scenario, meaning the current, the status of the state of the art right now, the use of consumable wearables in sleep and circadian research and in clinical sleep medicine is premature at this time and must be carefully evaluated on a case-by-case basis. And so we're getting close, but we're not there yet. And so let me unpack that a little bit. So first of all, there was a lot of discussion looking at accuracy. And when we look at the accuracy of a lot of the consumable wearables relative to what we see with the research wearables, the research grade wearables, they're both highly sensitive. And what that means is if we compare the assessment of whether or not it is sleep, and we compare the gold standard, which is again the brainwave recordings, the polysomnography, compared to the wearable, there's high sensitivity. So they both, when the gold standard says it's sleep, the wearable also says it's sleep, and that's greater than 90% of the time. But specificity with the consumable wearables is a concern because the proportion of time when the gold standard brainwave activity says it's not sleep, as we heard from Dr. Redline, the wearables overestimate sleep and they say it is sleep. And that sensitivity ranges anywhere from 20 to 80% for the, more than 80% for the research devices, but for the consumable wearables, considerably less, and on average about 50%. And so we have a need to go ahead and validate and improve upon the wearable technologies now, so that way we can develop wearables that are very useful in, again, large-scale studies in the field. And so this workshop white paper also discusses best practices for the validation studies and guidelines regarding how to choose a wearable device for research and clinical use. Next slide, please. Now, the existing research wearables do a number of things quite well right now. And so we can estimate here in these red boxes, total sleep time, how much wakefulness is occurring during the sleep episode when the people are lying there trying to sleep, as well as some estimates of sleep latency and sleep efficiency, the amount of time in bed spent asleep divided by time in bed. But there are many other outcomes, as Dr. Redline was discussing, and those are just a number of them are listed here, that wearables can assess, so that way we can get sleep in the home environment across many nights, weeks to even months, so we can get, again, a better assessment of the individual sleep. Next slide, please. And then when we think about circadian metrics and what to measure with wearables, the current wearables can get at light exposure, as I described earlier. Again, we can assess sleep latency, which is related to circadian timing, the timing of sleep, the timing of wake time, and the timing of physical activity. But there are many other outcomes in terms of circadian metrics that we currently do not have valid assessments for currently, and they're under development, and this again presents an opportunity. Next slide, please. Now, if we look at the current wearables, well, what do they assess? When we talk about sleep health versus circadian health, everything in red here in terms of sleep health, current wearable technologies are available to assess. So the duration of sleep, to estimate that, to estimate sleep timing, regularity, and quality. Whereas if we look at the circadian health outcomes, we currently do not have any wearable or biomarkers right now to assess circadian misalignment easily in the field. In terms of circadian timing, we can get at sleep timing, but circadian timing is something that is not accurately estimated currently. But again, we have those novel biomarkers that I mentioned that are promising. Circadian regularity, again, we don't have a way of assessing the circadian rhythm over many days to weeks. And again, that's an opportunity for the future, as well as assessments of circadian amplitude and the strength of the rhythm itself. Okay, next slide, please. So when we think about what are our opportunities right now, we do have an immediate opportunity to apply existing research tools to transform clinical and public health research. And so everything from what Dr. Redline and I have been discussing, looking at questionnaires, more specific detailed questionnaires or simple questions, the wearables themselves, these all can be added with other behavioral and physiological and clinical outcomes to go ahead and look at the population level, as well as in field studies, to integrate that with other omics, other biomarkers, cardiometabolic biomarkers, and cancer biomarkers. And so that opportunity is here now, and it will continue as we improve upon our wearable technologies. And so if we can collect objective sleep and circadian data, as well as subjective outcomes in sleep in these large-scale studies, this is going to allow us to have a better understanding of how sleep and circadian disturbances are contributing underlying mechanisms or worsening the pathophysiology of disease, as well as understanding when there's a tipping point where we transition from wellness to dysregulated physiology to eventually disease. Next slide, please. And it's becoming clear that understanding sleep health and circadian health is going to be an important component of personalized and preventative medicine. And the key thing here is timing. We need to understand the timing of all the assessments that we make now for health biomarkers, how that is timing relative to the sleep of the individual, of the circadian timing of the individual. And this is, again, going to help to improve the timing of personalized interventions. We know that, as we've talked about, both speakers here today, that there's bidirectional relationships between sleep and circadian rhythms, and pretty much every physiological system that you could think of in the body, including systems that are dysregulated in terms of metabolism and cancer. When we can utilize these tools in the future, they will also allow us to track adherence to treatment regimens, as well as to look at improvements associated with those treatment regimens. Next slide, please. And so then in terms of the future opportunities, we consider that these consumer wearables are, in essence, biomarkers themselves. And it's part of the biomarker space that is being developed by the field. Regardless of novel biomarkers, whether it's a blood sample to look at your sleep and circadian health, or these consumable wearables, both of these need validation against gold standards in the populations to be examined. And this is important to ensure not only scientific rigor of the research findings, but it's also very important that the information that is provided back to the public, whether it's public health messages or public information about their own health, about their own outcomes from these wearables, is accurate. So we avoid biasing the perceptions regarding sleep and circadian health if the information is not accurate. And we also have to recognize that there are potential ethical issues related to privacy and security when we consider efforts to link these wearable data with electronic medical records. And this is, again, especially important for these wearable technologies then to be accurate if, again, it is going to become part of the medical record, and how that is going to impact insurability, et cetera, in the future. Okay, next slide, please. With that, I'll hand it over to Dr. Redline.
Okay, thank you. So just a few concluding remarks. So I think we've heard the term biomarker a lot, and I just wanted to conclude by what is an ideal biomarker? And I wanted to emphasize the answer, as the answer is to many questions, it depends. And in this case, it depends on what you want to use it for. So for example, for screening, we usually need biomarkers that are highly sensitive to diseases that have high morbidity. For disease management, we need biomarkers that are highly responsive to changes in disease severity. For public health, we like to have biomarkers that will provide prognostic information and are highly specific. But moreover, we need biomarkers that are non-invasive in general, easily measured, inexpensive, provide rapid return of results. They can be derived from readily available sources. According to our needs, they have the appropriate sensitivity and specificity, and they have some link to the biology that we're interested in. Can I, oh, here we go. Okay, whoops, sorry, there's a delay here. Okay, so here is an example from one of my colleagues, Dr. Purcell, who's recently joined the sleep field and is already transforming it by really development of tools and asking great questions and doing fantastic analyses. So he points out that one of our, for decades long, low-tech biomarkers has been the height and weight nomogram. So anyone that's been to a pediatrician's office with their child knows that, these are, we provide some, that height and weight is routinely measured really straightforwardly. We usually feel that when we plot our child's height and weight, it's against population norms that we could interpret. And that in fact, by doing that, we could track our child relative to other children, that is percentiles, and track our child over time. Are they increasing or falling off their predicted curves? And that in itself gives information on their general health. But we know even this simple biomarker has limitations. The norms may not reflect the populations you apply it to, that there may be actually individual differences in growth trajectory that may not be reflected and not have prognostic significance. And when you give the information back to the families, they may not in fact understand what that information is. So it's interesting to take that framework and ask the question that Dr. Purcell has asked me and our group is, what would this look like for a sleep circadian biomarker? So I think that will be something we'll discuss, but I'd like to turn to maybe some efforts out, to actually use some of those scalable, and wearables and the types of data we get. So one recently published article used a fairly non-validated, but, in terms of precise gold standard, but used a wearable out there that was used on almost 70,000 adults between the ages of 1967 across the world and collected over 11 million nights of data. And you could see some graphs where sleep duration from this very large cohort is plotted by age and gender, weekdays and weekends on the top and bottom, and gender differences on this side. And what we're seeing is relatively tight confidence intervals at any given age. So there are some consistent inferences you could make. And what the study reported was that over age, nighttime sleep decreases, awakenings increase, sleep onset and offset advanced people shift earlier, and that there were gender differences. Men across ages sleep less than women, but women have more awakenings. A lot of face validity. But the question is, and so this may be a very, useful tool for tracking changes across populations and over time. The real question is how much measurement error is there, in that estimate of sleep duration and wakefulness to impact what we might want to, target for interventions or even interpret as responses in our clinical trials. And whoops, there's a delay. I'm sorry. As another example of the possibility of thinking about biomarkers is the opportunity to integrate across multiple data sources. So I want to highlight one of the data from Peter James, who has started to combine data from Fitbits, from a customized app that measured GPS as well as delivers contextual questions on the phone itself, for example, on stress levels and factors keeping people from sleeping, as well as from data from the cohort itself was used in. And this was used in the Nurses' Health Study in 435 participants that used this app and the Fitbit for 7 days and were asked to do this over 4 different seasons a year, resulting in 6 million participant minutes of heart rate, step data, sleep data, and location data. And what you're seeing in the lower corner here under GPS and the circle is data from one person over 7 days from those monitoring periods, with each day a different color. So you could see the GPS data give us information on where they locate, group space, in industrial space, movement patterns. And the circle gives us the 24-hour day with each line in colors showing, for example, the heart rate for each different day over the 24-hour clock, and the gray area showing the time the person slept. And you can see, for example, the dark gray is the consistent time per day that people slept, and the lighter grays show that variability. And this is just an idea of a sort of an integrated biomarker that really combines a whole host of contextual information and physiologic data at really low burden. I'm trying to move this. I'm sorry. Okay. I just, for time's sake, I want to say we could also relook at whether EEG, which we consider, we've thought about as being fairly invasive and requiring manual scoring, whether that can actually be used as a biomarker. So in a recent study that Dr. Purcell led that was just published, he looked at more than 200 sleep metrics in over 4,000 people that had EEG measured and actigraphy measured, as well as questionnaires. And of those 200 metrics, he identified 23 that were consistently associated with cognitive outcomes. And they did cluster in these domains we've talked about, from subjective sleep through macro and micro architecture and chronotype. But although there were associations with cognition, which was the outcome of this study, they all didn't predict cognition as well. And in fact, information on spindle and slow wave morphology, which is only obtained from the EEG, were really magnitudes more predictive of cognition than, say, sleep duration. So is that practical to think about? And so this is a delay. And I just want to say that now I'm going to skip. Now there are the open source tools that, and I give you a link to one of them that really allows some of this work. So to conclude, I wanted just to say there's a lot of challenges in front of us. One of them, is that we have to understand what we're measuring and why we're measuring. And in so doing, I think we can, choose the appropriate measurement properties. We also have to understand that although many of our tools may give highly correlated data, that sometimes the data may be unique, meaning they really truly are measuring a dimension, the other domains aren't. Whereas others may be proxies or surrogates really for the more refined measurement. And the latter might be able to overcome by sample size. So even if they're measuring similar things, but with differences in prediction, we still might be able to get the information we know. We again have to understand sources of biases and really understand how basic demographics influence norms. And finally, okay, I just wanted to end with the opportunities, and these reiterate what Dr. Wright spoke. So I think we really are at a point we have conceptual advances. We now have, I think, some exciting and developing frameworks to look at not just sleep or not just circadian disorders, their interactions, and thinking about data, not just from a single night, but multiple nights, multiple seasons, and integrating those data with key other behaviors important for metabolism and cancer, like diet and activity. We have the technological advances of really being able daily to gather big data from multiple sources of wearables and other sources, and the opportunity to embed those technical data into really our research platforms, our cohorts, as well as even in patient care. Thirdly, we have analytic advances. We now have the computational and statistical tools to integrate data, do dynamic modeling, and do machine learning. And even for the example I gave from Dr. Sean's Lunar platform, we can do objective processing and analysis of the EEG data to give reproducible and population relevant metrics. And then there's the opportunity to do really sophisticated designs where we may actually leverage big and small data sources, crude and refined, in order to make inferences that are most impactful. So thank you.
All right, thank you, Drs. Wright and Redline, for your excellent presentations. Now we'll open up the floor for a Q&A session. And as a reminder, please feel free to submit your questions using the chat or the Q&A box, and I'll ask these questions to the speakers on your behalf. And due to time constraints, I'll ask our speakers to limit their responses to these questions to about 1 or 2 minutes maximum. All right, so let's dive in with our first question. Many of the methodological challenges in sleep and circadian research are similar to those encountered in other behavioral domains, such as diet or physical activity assessment. What lessons, if any, or approaches from these other fields might be applied to sleep and circadian rhythms research? And we'll start off with Dr. Wright on this one.
Thank you, Dr. Connor. So I think, for example, I'll use the energy expenditure field as an example here. There are many tools that are available, and depending on the goal of the research, as Dr. Redline mentioned, you will select a tool that has the capability for your needs. So, for example, using heart rate to estimate energy expenditure, to looking at wearables on the wrist or on the hip to measure energy expenditure, all the way to double labeled water and whole room calorimeters. All of these have their advantages to be used in different populations and different scenarios, and they all have their limitations. And I think the same thing is true for all the biomarkers that we're discussing here for the sleep and circadian rhythms. And so we have opportunities. There are many tools in our toolbox to use, and then the question is which ones are best appropriate to be used. And in this case here, to utilize the tools from multiple fields in the same study, I think is really where we have an opportunity.
Thank you for that response, Dr. Wright. Dr. Redline, do you have anything to add to that question?
Yeah, the only thing I'd like to add is that I think the physical activity and nutrition field has done a fantastic job in understanding that despite limitations in knowledge at any given point, they've been able to utilize best knowledge at a point in time to make public health recommendations, and then willing to revisit and update those recommendations. And that is something I think that the sleep field can further embrace.
Excellent. Thanks for that response. All right, our next question from the audience is, could you comment more on the relative merits of different approaches to self-report of sleep, such as standardized survey questions about average sleep compared to 24-hour recall or diaries of sleep? And we'll start with Dr. Redline on this one.
Yeah, good question. So I think the questionnaire data is the data that's the easiest to get at, because it's usually 1 or 2 questions. So it's low burden. There, you may not be faced with missing data, and it asks people to reflect on their average experience, which may be very relevant. Diary-based data actually can give more granularity and maybe in some cases more accurate, but can be quite burdensome. And we often are faced with missing data and may take, a lot more, sort of work at cleaning up and understanding. For example, people don't seem to understand the differences between AM and PM, and that can be an issue. So those are just 2 examples. I just want to emphasize that self-reported data is important. Again, I said this before, I want to reiterate it. That's what is important to people, is their perception. And I think we should not throw out the importance of asking people how they perceive their sleep.
Thanks for that response. Dr. Wright, do you have anything to add about self-report, different self-report measures?
I agree, and I think that they're very useful, and I think they've led to a lot of understanding of the importance of sleep and circadian health in a lot of areas, including obesity and cancer. And so I think it has a role to be used, and now utilizing these new biomarkers in addition with that, I think we're going to learn even more.
Great, thank you. All right, we have a couple of questions regarding melatonin assessment that I'll address toward Dr. Wright. So the first question is, how often do samples of this biomarker need to be collected, specifically to account for its half-life? And does this frequency vary depending on the type of specimen collected? So blood versus urine versus saliva. The second part of this question is there a way to differentiate between natural melatonin levels in the body compared to the levels that might be derived from ingested melatonin supplements?
Great, thank you for the questions. So in terms of saliva and blood, those are the ones where we sample melatonin most frequently. And the majority of studies will sample hourly. But there are some studies, if they want to become even more precise, will sample every half an hour. And if we're trying to get the dim light melatonin onset, for example, in the home environment, we would start the sampling about 7 hours prior to the person's habitual bedtime and go 1 to 2 hours after habitual bedtime to try to capture that. When we're looking at things like urine samples to measure 6-sulfathione melatonin, there we're trying to get frequent urine samples. So let's say every 3 hours or so, you pull all the urine that is collected over 3 hour in 3 hour bins, and you measure it and take a small sample of that. And you do that across 24 to 48 hours to go ahead and get the entire rhythm. And that's been effectively done, like I said, in patients who are blind, for example, in the field. And then in terms of trying to distinguish between endogenous melatonin levels and if someone is taking a exogenous melatonin pill, well, we know that if you take a melatonin pill that is more than 0.5 milligrams or even above 0.3 milligrams, you're going to produce levels that are much higher than are produced endogenously. The melatonin assays currently cannot distinguish between the melatonin that's in that pill versus the melatonin that is endogenous. And so if someone does take melatonin and their levels are at your maximum of your sensitivity, it may be it's that high because someone has taken the pill. So we can't currently distinguish between those two.
Okay, great. Thank you for those responses. I think one final question that I'll direct toward Dr. Redline before we close out. Dr. Redline, you touched upon the many different aspects of sleep health that can be measured. If someone were starting a study right now and wanted to use a summary or composite measure of overall sleep health, what would you recommend for them to use?
Okay, so my answer was it depends. And again, it depends, I would really think about a combination of what the goal is as well as the resources and try to balance it. If in fact the idea is to try to get at a sleep health composite and there's limited resources and it's a preliminary work, actually Dr. Bicey has published and a very simple 6-item scale based on the RU sated that really asks people to, for each of those areas about regularity, timing, satisfaction, to rate each of those. So if there was limited resources, you want just get at a sleep health composite, that might be a place to begin, but it depends.
All right, excellent. Thank you both again to our speakers as well as to our audience for this great Q&A session. Unfortunately, we received a number of excellent questions today and don't have time to go and address them all, but we will plan to follow up on any of the unaddressed questions following the webinar. So now I'll hand it back to Dr. Shams-White for some closing remarks.
Thank you so much. And can we advance to the final slide? Thank you. So you were at the end of our webinar. Again, thank you so much to our speakers and audience for a really engaging presentation and discussion. As a reminder, a link to the event evaluation will be sent out shortly, so please do complete that when you have a moment to help inform our future webinars. And this webinar is also recorded and will be available to the public at a later date. And lastly, we'd like to share that our next webinar, The Challenges with Cancer, Obesity, and COVID-19, will be next Thursday, December 10th at the same time, 12 to 1:30. So with that, thank you again for attending, and I hope you all have a great day.